César Milstein Lecture, which is, of course, one of 4 named lectures that we have every year at LMB, where we invite people that have made really outstanding contributions to their area of research and beyond, and David very much fits that bill. So David studied in Chicago and then also went to medical school there. He then, I think, practiced as a clinician for a while, and then went to do basic research as a postdoc in the Whitehead Institute and MIT with Gerald Fink, and then started his own group at Harvard Medical School. So he's a Dana-Farber Cancer Institute professor, a professor of cell biology at Harvard Medical School, and he's been funded by the Howard Hughes Medical Institute since 2008, I believe. So his work really covers a broad range of areas related to cell division and chromosome segregation, highlights being mechanisms of actin nucleation and some of the consequences of aberrant cell division, including genome duplication events, and then more recently some beautiful work outlining a possible mechanism for the process of chromothripsis, which allows very rapid genome evolution. I believe we're going to hear about that today. So David's work's been recognized by a variety of different awards, most recently the Close Award for Outstanding Basic Cancer Research and election to the National Academy of Medicine last year. So David, we're very pleased to have you here and very much looking forward to your talk.
Thanks so much. I'm thrilled to be here. Super excited about all the people I'm going to meet today. So I'm going to tell you a little bit about the latter project that Joe alluded to. So yeah, my lab studies cell division, and we've over the years gotten very interested in the consequences of errors in cell division, which turn out to have a major role in shaping the architecture of cancer genomes. So we've been interested in how you go from here, nice genome with 46 chromosomes, to here, an extremely complex genome that could have hundreds, thousands, up to 10,000 rearrangements. And this actually is a process that we used to think was solved. So we used to think of it as just simple, classic Darwinian evolution. You get small scale alterations to the genome. They'd happen one after the next, and then you get from here to here eventually. However, you could just look at this genome. You actually can't see the complexity here. You'd have to sequence it. There are so many changes here, you could wonder how long would it really take to go from here to here if things just happen slowly and gradually. And so there's been a new idea that's emerged in the field. My group has been involved in, many people are working on it, which is that there are episodic saltatory events that drive extremely rapid evolution of the genome. And I'm going to talk today about one of them that Joe mentioned called chromothripsis. One important theme that's come out of this work is the central role of the architecture and the integrity of the nucleus in preserving the stability of the genome. What you're looking at over here is a rather extreme version of what's called cancer nuclear atypia. And nuclear atypia involves abnormalities in nuclear shape. These structures called chromosome bridges, these structures called micronuclei. So for almost 100 years, clinicians have been using the degree of nuclear atypia to predict patient prognosis, assign tumor grade, and choose therapy. Yet the actual functional consequence of forming these structures has been, in our view, a bit of an understudied problem and rather unclear. Okay, so today I'm going to focus on this process here. This is a neologism for chromosome shattering, first discovered by cancer genome sequencing by Peter Campbell at the Sanger Institute, and then subsequently found by many other groups and also found not only in human cancers, but also in congenital disease. And in principle, this kind of alteration can be transmitted through the germline. That's been shown in congenital disease, and therefore it, in principle, could contribute to organismal evolution in addition to its disease pathology roles. So chromothripsis is massive rearrangement of only one or a few chromosomes. And it's curious, how do you isolate and get so much mutagenesis on only one portion of the genome and leave the rest of the genome intact? And for other reasons I won't have time to go into, we've been studying the consequences of missegregating chromosomes. And we sort of hit on a very old problem, nicely reviewed in this work by Potu-Rao and Johnson and Sperling, that if you missegregate an intact chromosome or a large fragment of a chromosome, these chromosomes will wind up in their own nuclear structure, these micronuclei. They can also form bridges, which we think are essentially functionally equivalent structures. So we had gotten interested in the possibility that the missegregation of an intact chromosome would somehow lead to damage to the DNA on the chromosome. And we did a very simple experiment here in this 2012 paper. We just induced chromosome missegregation. We watched the micronucleus form, and then we sat back and watched, and to our surprise, what we got was abruptly, you would see the acquisition of extensive DNA damages, for example, marked by H2AX signal. The mechanism of this is I'm going to address today, but one of the key events here is that the nuclear envelope surrounding these structures is fragile and prone to spontaneous rupture. That was identified by Emily Hatch and Martin Hetzer. So what's going on here is that when the nuclear envelope ruptures, that's when you suddenly expose the chromosome to the interphase cytoplasm, and you get all this damage. So what intrigued us was that the damage is highly localized. It's only the chromosome that's in the micronucleus that got the damage, and all the other chromosomes look fine. So that made a lot of sense to speculate that this might cause chromothripsis. So a simple hypothesis was that an easy way to localize a lot of damage to one or a few chromosomes is just to physically partition them into their own nuclear structure, damage them, and then allow the broken chromosome to come back in a normal nucleus and get propagated. So it was a nice hypothesis, but our evidence was built purely on cell biology experiments. So to do a little bit better, we developed this approach, which we playfully call look-seek, which allowed us to generate missegregation, micronuclei, watch damage occur. Because the nuclear envelope ruptures, we assume that we wouldn't be able to actually see joining events that we could detect by sequencing. We'd have to wait until the next cell division when this chromosome or chromosome fragments wound up in a normal functioning nucleus with end joining system intact. So look-seek allowed us to basically do, I guess there are cell biologists here, it's essentially kind of like a CLEM experiment, but instead of using the electron microscope to increase the resolution of the light microscope, here we're using the genome sequencer. Now I'm just going to show you a little animation that summarizes our findings. So here we're going to start with the bad mitosis. We'll misattach chromosomes to the spindle. Those Some chromosomes will get caught in a tug of war. They'll lag at anaphase and then wind up getting partitioned into their own nuclear structure. Same thing can happen if you cut off the end of a chromosome. So a single break can generate this structure, which can then generate thousands of breaks. So these structures have fragile nuclear envelopes. They poorly import materials. That leads eventually to nuclear envelope rupture, damage, fragmentation. And then if the fragments sort of go willy-nilly and are divided between the two daughter cells, you'll wind up with this kind of sequence feature. So here you're just looking at the sequence, you're looking at the haplotype of the homolog that was in the micronucleus. Happy to take questions on how we do that. But what you can see here is a oscillating copy number pattern, which is the characteristic signature of chromothripsis. So the picture is dominated by having fragments retained, loss, and very satisfyingly, when we compared the two daughter cells, we saw a mirror image copy number pattern. So if a fragment was distributed to one daughter, it was missing in the other, present in the other, missing in the first, et cetera, et cetera. So this mirror image pattern essentially proved that the chromosome was shattered. Okay, and in addition, if you can shatter a chromosome, the fragments can ligate together. There's no particular reason that they couldn't also ligate and form circles, and we could actually see that very nicely in our sequencing experiments. So we thought we were kind of looking at the birth of extrachromosomal DNA circles, which I'm sure everybody here knows are important vehicles for amplifying oncogenes. Okay, so this slide is just to hopefully convince you that there are a lot of really interesting fundamental questions buried in this pathological process. What goes wrong with nuclear envelope, and why do you get fragility and rupture? The main things I'm going to talk about today are the chromosome fragmentation mechanisms. And what we discovered in a 2020 paper is that the fragmentation is happening in two waves. One in interphase as soon as the envelope ruptures, and then there's a whole second wave that happens in mitosis. And today, in the last part of my talk, I'm going to discuss new progress on understanding how that works. Okay, so I'm doing a little history wrap-up. I'm just going to show you one slide that illustrates that one of the major reasons that the nuclear envelope is defective on these structures is that they have a very striking defect in nuclear envelope assembly. So this is anaphase, telophase chromosomes. That's a lagging chromosome. And what you can see here is that some nuclear envelope proteins assemble perfectly normally, both around the main chromosomes and the lagging chromosome. But here you're looking at the nuclear pore complexes. You can see that there is a very striking depletion of nuclear pore complexes on these lagging chromosomes that will eventually become micronuclei. So we think this might be sort of the original sin of the process, the first defect that leads to envelope fragility. Okay, so I'm going to quickly review some of our work on the interphase pathway and give you just a quick update on things we're doing now, rather preliminary, and then I'm going to focus on mitosis. Okay, so I'm going to tell you, just summarize a story where we found that a pathological version of DNA base excision repair is responsible for a substantial portion of the damage when the nuclear envelope ruptures. So just to remind you, during normal base excision, repair, you have modified bases that are recognized by enzymes called glycosylases. And the glycosylases remove the damaged base and leave a so-called abasic site. Then the APE1 endonuclease generates a nick. And then typically, with very high efficiency, these undergo high-fidelity repair. However, if repair fails, you can wind up with double-strand breaks. That's illustrated here. That can happen either by APE1 making another nick close by on the opposite strand, or by DNA replication passing through the nick. Okay, so now I'm just going to summarize work from Changmin Tang, and I'll show you just a snippet of data supporting some of the conclusions. So the process starts with a massive accumulation of RNA-DNA hybrids on micronuclear chromosomes after the envelope ruptures. Inspired in part by elegant work from Peter Beal's lab that biochemically showed that ADAR enzymes, which are normally regulating innate immunity and deaminating adenines on the basis of double-stranded RNA, what Beal's lab showed was that on hybrids, the ADAR enzymes not only can deaminate the adenines on the RNA strand, but also the DNA strand. So we found that on micronuclear chromosomes, indeed, there is a lot of deamination of adenines and the formation of deoxyinosine. That recruits a specific glycosylase called the N-methylpurine DNA glycosylase, or MPG. And MPG then removes the hypoxanthine base of the inosine residue. That leads, of course, to single-stranded nicks and ultimately double-stranded DNA breaks. So I'll just show you a little bit of data supporting this. So Changmin purified micronuclei and the primary nuclei from the same cells. And here what he did is just a simple dot blot assay with an antibody that recognizes RNA-DNA hybrids. What you can see is that there's a huge accumulation of these hybrids on micronuclear chromosomes that's insensitive to RNase A, but quite sensitive to RNase H1, which digests hybrids. It's known that the antibody has some propensity to cross-react with double-stranded RNA, and if we included RNase 3, we essentially completely got rid of all the signal. Okay, and this is just some images with endogenous antibodies or exogenous tags, and you can see that this MPG glycosylase accumulates very strikingly on the ruptured micronuclei. So that was sort of a comprehensive mechanism that drives some of the damage in micronuclear chromosomes. However, that accounts for about 60% of the interphase damage, and more recently, several other mechanisms have also been proposed. So my postdoc, Tejinder Ubi, who just recently joined the project, decided to take this on and try to sort out how all the various proposals work together. And the first thing that she did was to set up a simple automated image analysis pipeline where she used AI-based methods developed at the Broad Institute to segment cells and measure damage in micronuclei. You can see here that worked very nicely. So these are 3 MPG knockout clones, 3 APE1 cells, and you can see that there's a substantial reduction in DNA damage. Moreover, consistent with the idea that this is really one pathway, these effects were epistatic, so you can make double mutants, and you got basically the same result. So now that we can, in a nice automated way, detect damage in micronuclei, what Tejinder set up is a technique developed by Paul Blaney at the Broad Institute called optical pooled screening. And what this does is allow you to do a CRISPR screen and directly relate the genetic perturbation to a morphological phenotype that you're interested in. So you add the CRISPR library. In our case, we induced the formation of micronuclei. We measured DNA damage, and then the trick is to associate the guide sequence for the RNA that caused the phenotype with the actual phenotype. And this is accomplished by a process of rolling circle amplification in situ of the guide sequence. That's followed then by Illumina sequencing performed directly on the image cells. And this is all new data. We're just sorting through and validating hits, but the screen looks promising. Our top hit was H2AX, which is good. That's what we use to score damage in the screen. And right near the top is MPG. Okay. Now I'm going to turn to addressing the question of how the chromosomes get shattered in mitosis in the second wave of damage. So this is a bit of data from Neil Umbright's paper that illustrates the damage that occurs in mitosis. So I told you if the nuclear envelope ruptures an interphase, you get a lot of damage. If you make it to G2 and the nuclear envelope is intact, you have very little damage, shown here, and also very little DNA replication. However, if you synchronize or release these cells into mitosis, and you come back 20 minutes later, you see a striking result. There's a burst of DNA replication happening in mitosis, and that's coupled to a very large amount of DNA damage. Okay, so I'm going to try to convince you that that damage that you see there is coming from the corruption of a normal repair process called common fragile site expression. So common fragile site expression is a process that's triggered by DNA replication stress. And if replication is not completed, cells can, with a small number of unreplicated sites, can make it into mitosis, even with unreplicated segments of DNA. And this typically happens at difficult to replicate sites in the genome, which are called common fragile sites. So I hope you can appreciate here that this is kind of an emergency because mitosis is short. And mitosis is short, and so you have to do something with this unreplicated DNA. So you either need to complete replication, or you need to break the strands and allow chromosome segregation. So common fragile site expression has been known for quite a long time, and it was first defined as being visible breaks and gaps on mitotic chromosomes induced by replication stress. Well, common fragile site expression is, the mechanism underlying this is much debated, but there's a general appreciation that the process involves breakage of DNA and some form of mitotic DNA synthesis. Whether that's a conservative mode of break-induced replication synthesis or a normal S-phase synthesis has not been that clear. So we kind of view the largely unreplicated segments of DNA on micronuclear chromosomes as a collection of fragile sites. And that's because they replicate extremely inefficiently and then enter mitosis with a large number of stalled forks. So even if you have a few stalled forks, you can imagine that you can fix them and ligate them properly. But if you have many, you could see how the system could become overwhelmed, and then you would get lots of chromosome breaks. You wouldn't have the ability to keep track of them all. You'd randomly combine them and get chromothripsis in principle. So there's been elegant cell biology and genetic analysis of common fragile site expression, but the field has lacked a biochemical system to actually do mechanistic analysis. And to address this, I initiated a collaboration with my colleague Johannes Walter at Harvard Medical School, and Johannes uses Xenopus egg extracts to study replication and repair. And we thought, well, what would happen if we had stalled forks and then we had mitotic cyclin CDK1? What would happen to the forks? And this initial paper was published a while ago, and in that study, we identified a major role for an E3 ubiquitin ligase called TRAPE in triggering the breakage of mitotic forks. TRAPE is an interesting molecule. During interphase, its activity is highly constrained. It seems this is mostly work from Johannes' lab, but a number of other labs have also contributed, including Karim Labib. So TRAPE appears to be constrained at interphase, and it seems to be oriented on the replosome in a way that its ring domains are oriented such that you cannot ubiquitinate the CMG that it's associated with, but you can ubiquitinate barriers, proteins, DNA protein crosslinks in front of the replosome. So you can think of TRAPE as being an interface, kind of like an antenna that's looking for barriers to replication fork progression. So something really different and interesting happened in mitosis, which is in, or mitotic extracts. So what we did here to model common fragile site expression was to take a plasmid with lac repressor arrays, add cyclin B CDK1 to drive you into mitosis-like state, replicate the plasmid, and then with the barrier in interphase, you get very stable stalled forks that just sit there. But when you add cyclin B1 CDK, something different happens. So this is what happens in interphase. So here we're replicating the plasmid. You can see you start with some replication intermediates and then mature plasmids with no barrier. You add cyclin B CDK1, you basically get the same result. Actually, replication goes a little bit more efficiently, presumably because CDK activity fires more origins. So this is what happens in interphase extract. You get this stalled forks, you get this so-called theta structure. You can cut these out and do EM and see that it's matching this structure here. But when you add cyclin B CDK1, something really remarkable happened. Instead of replication completing, what's happening is that the stall forks are breaking, and then they're getting ligated together by a polymerase, DNA polymerase theta, and they're kind of making a massive structure that won't even enter the gel. So this is I'd propose is at least reminiscent of what happens in chromothripsis, because you're getting shattering and the whole mix is getting ligated together. Okay, so what we found was that in mitosis, TRAPE gets reprogrammed in some way such that it can now ubiquitinate its host replicosome. And that triggers the stripping of the whole replicosome, CMG, and then the rest of the replicosome by the P97 ATPase. So this leaves exposed DNA, and that exposed DNA that had previously been encircled by CMG then becomes available when CMG is unloaded, and then you get fork breakage. And here we come to sort of our hypothesis. What we're hypothesizing is that it's probably the leading strand templates that become available. So you're selectively breaking only the leading strand templates. And if that's the case, you can make a very appealing story of where that goes. Because first of all, you'll get one chromatid here that can be patched up with just a little DNA synthesis and repair. So maybe this is the mitotic synthesis that we were looking at in the first slide in this section that I talked about. That would then be followed by joining of these two ends here, which would generate a small deletion. So you can see that what's happening here is that you form a deletion, but you're also swapping the arms on the sister chromatids, the so-called sister chromatid exchange. And this is appealing because the two major features of common fragile sites that have been identified are small deletions, this is all over cancer genomes, and sister chromatid exchange. And there have been other models proposed, but they don't explain these sort of hallmark features of fragile site expression. So more recently, we've identified a mechanism that explains how TRAPE becomes reoriented in mitosis, enabling wholesale removal of the CMG from all mitotic chromosomes. So this started with my postdoc, Archana Krishnamurthy, who did a plasmid pull-down experiment where she replicated plasmids in the presence of the barrier, and then used P97 inhibitor, that's the ATPase that's stripping CMG off the chromosomes, to kind of lock the stall forks in place so that they didn't break and get processed. And then she performed mass spectrometry looking for proteins that accumulate on the stalled mitotic fork specifically. And one of the proteins that she identified is an ATPase called TTF2. So TTF2 is an excellent candidate to have a mitosis-specific function because it's cytoplasmic in interphase and only gains access to chromosomes in mitosis. Furthermore, it has a known mitotic function, which that via its ATPase domain, it clears RNA polymerase II from mitotic chromosomes. That appears to probably be its essential function. So we used AlphaFold 3 to look for interactions between a whole group of proteins involved in replication and repair. And we identified an interaction between TTF2 and this E3, a segment of this E3 ligase called TRAPE. What you can see here is that this is binding in a basic pocket on TTF2 on a zinc finger, and if it was phosphorylated, you get nice charge complementarity. So you have a specific interaction between this CDK phosphorylation site and the zinc finger domain of TTF2 on residue lysine 16. So I'll just walk through some of the data that shows that this phosphorylation is actually triggering a reorientation of TRAPE on the mitotic replosome. So this is TRAPE. Here's its ring domain. And the critical CK phosphorylation site that we identified is here. We made a phosphospecific antibody, and you can see that it nicely regulates in vitro a fragment of TRAPE. You can see it gets phosphorylated if you have cyclin B CDK1, and you abolish that phosphorylation if you mutate that site. So, and this mitotic phosphorylation, if we took a peptide with the phosphorylated site and then in place, and then we added cyclin B CDK1, you can see that TTF2 will now pull down this straight peptide in vitro in a CDK phosphorylation dependent manner. And if we mutate this lysine residue on TTF2, you lose this interaction. Okay, so now I'm going to show you that that phosphorylation is actually functionally important. So what you're looking at here is, again, we're using the plasmid pull down in extracts over time. And what you can see is that initially you pull down CMG subunits that are ubiquitinated, shown here. Again, we're in the presence of the P97 inhibitor, so we can accumulate these structures. And you can see that C45, one of the key replosome components of the active CMG, is initially on the stall fork, but both of these are rapidly lost. However, if we get rid of TRAPE, both CMG proteins here, you can see that they're stably associated with the stall fork. If we add back wild-type TRAPE, we restore function. And then if we mutate this CDK phosphorylation site, we greatly attenuate the unloading of CMG. Moreover, if the only thing that TTF2 is doing is to provide a link to the replosome, you would think that we would be able to take just this sequence from TTF2 and engraft it onto TRAPE, and now you should have phosphorylation independent function that bypasses the whole requirement. I didn't have time to show you the data, but the other interaction partner for TTF2 is the Pol E2 subunit of DNA polymerase epsilon. Okay, so this is the construct. So here we put in the Pol E2 interacting peptide from TTF2, and we mutated the CDK phosphorylation on TRAPE with the hypothesis that now you should become totally independent of that. And then here in a TRAPE TTF2 double depletion, you can see that CMG unloading completely fails. Now you add back the fusion and you significantly, maybe a little bit partially, rescue the effect. Okay, so TRAPE phosphorylation, I don't have time to show you the data, doesn't affect its interface functions such as interstrand crosslink repair, DNA protein crosslink repair. Okay, so we'd like to propose a mechanism for reorienting TRAPE on the mitotic replosome that allows for this special function to deal with unreplicated DNA that slips into mitosis. So the proposal here is that the phosphorylation site binds the TTF2 zinc finger, and this is in an otherwise unstructured region of TTF2, and there's a short linear motif that binds Pol E2 from the region of TRAPE that's involved. So simple linear bridge here. And I'm not going to show all the data, but we can mutate both sides of all of these interactions, you get the same result that I just showed you. All points of contact in this molecular bridge are required for CMG unloading and fork breakage and mitosis. So just to wrap up here, we think this is not only an interesting mechanism for dealing with unreplicated DNA and mitosis, but it, I think, highlights an interesting general concept that in mitosis, the replisome actually gets reorganized to allow this regulatory activity that's specific to mitosis. Okay. So this work is posted on bioRxiv. There's a companion paper from Karim Labib. I just want to stop and thank members of my lab, shown here, all masked up. And this is folks in Johannes's lab. And I want to highlight that a joint trainee postdoc, Gailani Khan, is sort of the hero of this story. So thank you for listening. Happy to take questions.
Great. Thanks very much, David. Questions? Julian.
Going back to the very early burst of RNA formation or RNA DNA hybrid formation in nuclei with a ruptured template, what do you think is driving that? What structure do you think is driving topic transcription?
So we know that we can block it with DRB, which also is blocking the damage. So what we presume, we know from EU incorporation in intact micronuclei, that transcription already starts out very defective. And then it abruptly stops as soon as the nuclear envelope ruptures. So we hypothesize that the simplest explanation would be that you've got pokey transcription to begin with, and that when transcription abruptly stops, the RNA would simply lay down on its template strand.
I'll buzz over there.
Thanks for a brilliant talk,
Hey,
David.
Buzz.
Just quickly, you sort of allude to the idea that maybe this could have a role in evolution. And I was wondering if you look across evolution, are there instances where a single chromosome has been, where there's evidence of a single chromosome being shattered in a sort of in an evolutionary context?
I've talked to a lot of people that do this professionally, and I don't think there are examples we can identify specifically that address your question. But, you know, in evolutionary time, there's a lot of things that could cover the traces of this kind of event. There are examples in evolution where many chromosomes seem to be shattered relative to proximal ancestors. So that raises the possibility that something like this could contribute. And chromothripsis is not specific to humans or diseases. It also occurs in plants. It also occurs by a mechanism involving micronuclei.
Okay.
Okay, thank you. So very interesting talk, and I was just wondering during the micronuclei formation or the DNA breaks, why didn't the cell activate p53 and induce apoptosis? Why the cell can survive?
Okay, that's a good point. And you hit on something that I didn't tell you, which is that we knocked down p53 so we could see these events. So in general, the manipulations that cause micronucleation will activate p53. We don't think it has anything to do with making a micronucleus. Most of the manipulations that you would make will prolong mitosis, and it's well known that prolonging mitosis will activate p53. But I think another interesting question is whether, why don't you activate the DNA damage checkpoint and then stop cell cycle progression? And there's actually some really interesting studies, particularly an early study in yeast, that shows that the DNA damage signal is actually limited to the nucleus where the damage occurs. So if you make a yeast that has 2 nuclei, you can do this with various genetic tricks, and then you make a break in one nucleus. That nucleus will not divide, but the other one seems not to know about the damage and goes on its merry way and divides.
Hey David, amazing work. So I have a very naive question. So during the segregation of these micronuclei, is there any selection of what complexes or what machineries from the nuclei, the parental one, that gets segregated into these nuclei, or just completely random?
I'm not sure I fully understand the question, but I don't think I think it's an accident, so I don't think there's anything very programmed about what happens to the chromosome, like after the nuclear envelope ruptures. In terms of selection, I think the chromosome from the micronucleus, when it's sitting in the cytoplasm, doesn't express anything. So it changes phenotype because you essentially have a whole chromosome that's not functional, so you have kind of a pseudo-aneuploidy. But of course, once chromosome's fragmented and things can come back into the normal nucleus, then you can come under selection, of course.
I was curious, so after the chromosome fragments, is there some mechanism to gather it back together into the reformed nucleus during mitosis, or so are all the fragments accounted for in the 2 daughter
Yes.
cells? And if so, what would that mechanism be?
Yeah, that's a really great question. Yeah, so this is work from others, mostly Dan DeRocher. There's a complex between CYP2A, a protein called CYP2A, and a protein called TOP-BP1. And what's really interesting is that this complex seems to make some kind of linear, speculated to be filament that is coating the broken chromosome. So that is sort of holding Humpty Dumpty back together again, at least for a while. But it's very inefficient. So if we sequence 10 pairs of cells, we would see that all the fragments are going to one daughter cell in 6 of them. And the remainder of them, you get split between the 2 daughter cells.
Question just behind David.
Yeah, sorry. Yeah. I was quite interested if you had any. Sorry.
Sorry, go.
I was interested to hear what your thoughts are on what happens to kind of transcription when you remodel the replosome and you kind of go into mitosis, evicting replication forks, and how that kind of interplays, especially when you initially have these DNA RNA hybrids and conflicts.
Yeah, the simple answer to that is that we haven't looked directly at that. I think it's an interesting question. We did have a study in 2023 where we showed that when you have a micronucleus that ruptures in the cytoplasm, you acquire altered chromatin marks, particularly accumulate a lot of repressive marks. And then we used a variation on look-seq to show that even when that chromosome comes back in a normal nucleus, that you still have a heritable changes to transcription. So in addition to the genetic changes, these events also have the potential to alter the chromatin state.
So my question is around the P53 question, which is, we know that the cancer cells are aneuploid and chromosome aberrations are frequent there. So do you think that most of them, the cancer cells, first have to acquire a P53 deficiency or a homologous recombination deficiency to become aneuploid and to acquire chromothripsis?
I think they're strongly, if they lose P53, they're strongly favored to undergo this kind of thing. However, the human patients that have congenital disease due to chromothripsis are p53 proficient. So you can get chromothripsis with a whole human that has this in every one of their cells. So I think p53 is loss is facilitating, but not, determinative. David?
You mentioned that the shattered chromosomes are mirrored.
Hmm?
You mentioned that the shattered chromosomes are mirrored in the chromosomes.
The pattern of the copy numbers.
So what happens to the centromeres and the telomeres between the two pairs of shattered chromosomes? Are they shared equally? And what happens when you have a loss of telomere?
I'm a little embarrassed to say that we haven't looked at that. I've been trying to get somebody in the lab to look at that. I mean, there's kind of a null hypothesis that's not that interesting, which is that it's just DNA, everything's getting shattered, so you'll see exactly the same thing. So that's I haven't been able to overcome that argument to get that experiment done.
Hi, thank you I thought that was really interesting. I guess you may have explained this, but why is it in your first division when you get the lagging chromatid, you get a micronucleus forming around that, but then in the next one, the fragmented nuclei get included in the regular nucleus and don't form a bunch of micronuclei again?
Well, they can. So you can form a micronucleus in one generation. It can seem to reincorporate in the next, and then not get fully repaired and then pop out in subsequent ones. So, I mean, the fraction of them that really become functional chromosomes is not clear. I would guess that almost all of these events are terminal events for the chromosome that was involved, and that you don't reassemble a functional chromosome. But obviously under selection, sometimes you can.
When these kind of broken and recombined chromosomes get reincorporated back into the nucleus, do you see pervasive changes in transcription as a result of changes of, like euchromatin, heterochromatin, where these genes localize?
Yeah, a great question. I was alluding to this 2023 paper that we had where we looked directly at that question. So at least initially, and then we also did long-term evolution experiments where we could see at least what we think to be the footprint of these early events. Initially, what you have is a very large-scale suppression of transcription. It's variable and variably penetrant, but it also, that's interesting because that gives you variation in gene expression. And the mechanism of that is not clear, but in our paper and other preliminary results that we have, it looks like the assembly of very interesting kind of mysterious nuclear bodies called 53BP1 bodies are kind of critical for getting that transcriptional suppression.
Buzz got another question.
Yeah, so I should probably know this, but if you have large numbers of copies of chromosomes that get missegregated or get lagging, is there a critical number at which you then can assemble a proper envelope with enough pores, or is it really localization and polarity of things like Aurora that are setting that up?
Yeah, so we don't think it's chromosome number. You can get quite a variable number of chromosomes involved. And there's some very interesting tumors where for some reason you mostly get almost all of the chromosomes involved. But what we think is going on is that it's all about the spindle providing a barrier to access of membranes that are competent for nuclear pore complex assembly. So, in one style of nuclear envelope assembly is to have large sheets with little holes in them that are kind of landing pads for the nascent nuclear pore complexes to assemble. And those sheets don't gain access to the central part of the spindle. I should mention that there's another style of doing this that was discovered by my former postdoc, Jing Zhao, And Jing just started her lab here. She's a former LMB grad student, and she just started her lab in more than half a year ago over at the Cambridge Institute for Medical Research.
Laurie?
I just wondered if you could say a bit more about TTF2. So you think it's only acting as a tether and no other function of, because it's also an ATP.
Okay, so it has these two domains. The ATPases clearly has an essential role in probably clearing RNA polymerase II. It could do other things that we don't know about. And the N-terminus seems to function autonomously. So you can delete it, you're defective in the sort of fragile site response part of it, but you're alive because you have the ATPase. In vitro, the N-terminus is sufficient for the response to unreplicated DNA. So why those two functions are linked together in an evolutionary conserved manner, I think is a really interesting question, but we really don't know about it.
That's great, thanks. I was just trying to understand why you get the chromosome fragmentation in mitosis. And is that because you have an elevated frequency of these common fragile sites? Or is the CMG removal pathway or mitotic DNA synthesis somehow compromised in those micronucleus?
I mean, for sure, we think that we know that you enter mitosis with huge amounts of unreplicated DNA. We do know that replication is initiating, so you have a large burden of these, junctions that would be cleaved. So I think that's a major part of the story. Whether there are other things going on, that's a little harder to say for sure, but could be.
Okay, great. So if there's no further questions, we can just thank Dave again for a great talk.